Okay, so you've brought us a whole bunch of stuff on good laboratory practices, GLP. Looks like we've got regulations, guidance on GMP, and even some broader discussions on quality in the lab. This is going to be a deep dive.
You got it. We're going to really break down what GLP is all about, why it's so crucial in non-clinical testing, and how it all ties into cGMP compliance down the line.
Sounds like a plan. We want to make sure you're not drowning in technical jargon, but that you walk away really understanding the core principles.
That's the goal.
So we'll be hitting the key points, like what exactly is GLP? How does it connect with cGMP? They're different, right? But they must be related somehow.
Oh, they're definitely connected.
And then there's the whole thing about standardized procedures, making sure our testing methods are super solid through validation, the why behind those strict environmental controls, the importance of documentation, documentation, documentation. And of course, keeping everyone trained up.
All essential pieces of the puzzle.
Okay, so first things first, what are good laboratory practices?
You can think of GLP as like the quality control system for the science behind medicine. It's all about how we plan, do, monitor, record, report, and even archive those non-clinical safety studies.
The studies that happen before we even think about testing in humans.
Exactly. We need to be absolutely sure that the data from these studies is accurate, reliable, totally trustworthy.
Because these early studies are what tell us if a drug is even safe enough to consider testing in people, right?
Exactly. And that's where the connection to current good manufacturing practices, or cGMP, comes in. You see, cGMP focuses on the manufacturing side, making sure every batch of a drug is produced to the same high-quality standard.
So cGMP kicks in once we know a drug is promising and we're ready to start making it on a large scale.
Exactly. GLP is about those early non-clinical studies, things like toxicology and pharmacology, the foundation of the drug safety profile.
I see. So GLP makes sure the science is solid, and then cGMP makes sure the manufacturing process keeps up that same level of quality.
That's it. GLP is like checking if a compound even looks safe, what its potential effects might be, and then cGMP is all about how to produce it reliably if it passes those initial safety checks.
That makes sense. So GOP first, then cGMP. But let's go back to those standardized procedures you mentioned. What does that actually look like in a GLP lab?
Well, that's where things get really concrete. We're talking about standard operating procedures, or SOPs. These are like the super detailed instruction manuals for every single thing you do in the lab.
So every scientist knows exactly how to do each test the same way every time.
Right. You wouldn't want everyone doing things differently, would you? That would throw in a ton of variability, and you wouldn't be able to trust the results.
So it's like having a precise recipe for every single experiment.
Exactly. Our sources really emphasize this. You need an SOP for everything, how to operate and maintain your instruments, how to calibrate them, everything.
And I'm guessing there's a lot of sophisticated equipment in a GLP lab.
Oh, yeah. Take high-performance liquid chromatography, or HPLC. It's used all the time to separate and quantify different components in a sample. It's a workhorse. But you need a detailed SOP for it to make sure everyone uses it correctly.
I see. So it's not just about knowing how to use the equipment, it's about making sure it's performing correctly too.
Right. We call that qualification, making sure it's installed correctly, that it's operating as it should, and that it's calibrated regularly for accuracy and precision. And it goes beyond the fancy equipment, too. You need SOPs for routine things like cleaning and sanitizing, even pest control for the whole facility.
Because even a tiny contaminant could mess up a sensitive experiment. So we need SOPs for everything, and we have to follow them strictly, and then we have to document that we follow them.
You got it. The Easy Global Training transcript makes that clear. If it's not documented, it didn't happen.
And that's a big deal for the FDA, right?
Huge. They really focus on written procedures and lab practices. It's one of the most common things they cite on those Form 483 observations. They need to see that system in place, see that it's being followed, and have proof.
Okay, so SOPs are all about consistency, but how do we know that the tests themselves are actually giving us good data, that they're accurate and reliable?
That's where method validation comes in. It's a core part of GLP. Think of it as proving that a test method does what it's supposed to do, that it's going to produce trustworthy results.
So we're basically testing the test itself.
Precisely. And there are a few key things we look at during validation. Accuracy, for example. Does the method give us results that are close to the true value?
So if we know there should be a certain amount of a substance in a sample, does the test accurately measure that amount?
Right. We often use spike samples to check this, where we add a known amount of a substance and see if the test can measure it correctly.
Makes sense. And what about precision?
Precision is all about consistency. If we run the same test multiple times on the same sample, do we get similar results each time?
So it's about how repeatable the test results are.
Exactly. And then there's specificity. We want to make sure the test is only measuring what we want it to measure, that there's no interference from other things in the sample or from the instruments themselves.
Because any interference could throw off the results.
Exactly. And finally, there's ruggedness. This is about how well the method holds up under slightly different conditions.
So if a different person runs the test or they use slightly different equipment, do we still get reliable results?
Right, it's like making sure a recipe works even if a different cook follows it. A rugged method is one that can handle some variation without messing up the results.
Makes sense. And I think the transcript mentions some resources like USP and AOAC. What are those?
Those are organizations that develop and publish scientifically validated test methods. Labs can often use these methods or adapt them for their own GLP studies.
So it's like having a pre-approved set of instructions that we know are reliable.
Exactly. But even then, we still need to do our own verification to make sure it works in our lab with our specific equipment and samples.
Okay, so we've talked about SOPs, method validation. Now let's move on to the lab environment itself. Why are those strict environmental controls so important?
Well, think about it. The conditions in the lab can have a big impact on our experiments and samples. We want to minimize any chance of contamination and make sure environmental factors aren't influencing our results.
So it's about keeping the lab super clean and controlled.
Exactly. And the GMP regulations are very specific about this. The lab needs to be the right size, the right construction, and even in the right location. It all needs to be designed to make cleaning and maintenance easier.
I see. It starts with the design of the building itself.
Right. There needs to be enough space to prevent mix-ups and contamination, and the flow of people and materials needs to be planned out carefully. You don't want to be carrying potentially contaminated things through a clean area, for example.
Makes sense. And the transcript mentions something called hygiene zoning. What's that about?
Hygiene zoning is all about creating different levels of cleanliness in the lab. Some areas need to be super clean, like where we do aseptic processing.
That's where we handle sterile materials, right?
Exactly. And those areas need smooth, hard surfaces that are easy to clean. But other areas might not need to be quite as strict. The point is to create a clear separation between areas with different levels of cleanliness.
So you're basically controlling the flow of contamination.
Right. And then there are all the other controls we need to think about. Ventilation, air filtration, controlling air pressure, monitoring microorganisms, dust, humidity, temperature.
So many things to keep track of.
It's a lot. And of course, we have to handle lab waste properly, treat it, and dispose of it safely to prevent contamination.
It sounds like a huge job just to maintain the right environment.
It definitely is. And don't forget about equipment qualification. We need to make sure that all our instruments are not just working, but working correctly in that controlled environment.
Because a malfunctioning instrument could give us bad data, even if the environment is perfect.
Exactly. So it's all connected. Okay, so we've talked about control environments, validated methods, standardized procedures. Sounds like a lot to keep track of. Which brings us to documentation, right?
That phrase keeps coming up. If it's not documented, it didn't happen. Why is documentation so important in GLP?
Because it's the proof. It's the official, permanent record of everything that happens in the lab. It's how we show we're complying with regulations. It's how we reconstruct a study if we need to. And it's how we ensure the integrity and traceability of our data.
So every step needs to be recorded in real time.
Yes. And there are specific rules for how to do this. We have to use permanent ink, no pencils. All entries need to be legible. And corrections have to be done a certain way.
I bet there are rules for how to correct a mistake, too.
There are. You can't just erase or white it out. You have to cross it out with a single line, initial, and date it. And the original entry has to still be visible.
Wow, that's meticulous.
It has to be. We also need to have detailed records for each lab notebook, unique ID numbers, who it's assigned to, a table of contents, any deviations from procedures, unexpected observations, problems that arise, they all need to be documented too.
Along with what happened and what you did to fix it.
Right. And of course, every entry needs to be signed and dated by the person who did the work.
Talk about a paper trail.
Or an electronic trail if we're using electronic systems. But even then, the software needs to be validated, and there have to be audit trails to track every change. And signatures have to be electronic, but still linked to a specific person.
It's about making sure everything is attributable, so we know who did what and when.
Exactly. And we need those detailed logbooks for equipment, recording every calibration and maintenance activity. And then there are batch records, which track everything about a specific batch of product we're testing. All the components used, the controls performed, the yield calculations.
It really is documenting everything.
And it's not just about the paperwork. We need to be able to clearly identify every single sample.
So you can trace any piece of data back to the exact sample it came from. It's really impressive, the level of detail and rigor involved. And of course, none of this would be possible without the people doing the work. Which brings us to training.
You got it. Well-trained personnel are essential for GLP. Everyone needs to be trained on the procedures they're doing, the regulations that apply to their work, and good documentation practices.
So it's not just about knowing how to run a test, it's about knowing how to document it properly, too.
Right, and training isn't a one-time thing. It needs to be ongoing. We have to keep our skills sharp, learn about any changes to procedures or regulations, and really foster a culture of quality in the lab.
It's about continuous improvement. So let's pull it all together. How do all these pieces, SOPs, validation, controls, documentation, training, work together to ensure that our non-clinical data is reliable?
They all build on each other. FOPs provide the framework for consistency. Validation ensures the tests themselves are reliable. Environmental controls minimize external factors that could affect results. Documentation provides the proof that everything was done correctly. And training makes sure everyone is competent and doing things the right way.
And that reliability is what the FDA relies on to make decisions about drug safety and efficacy.
Absolutely. This data is the foundation of regulatory submissions. It's what helps ensure that the medicines that reach the public are safe and effective.
And if a lab doesn't follow GLP, there can be serious consequences.
Oh yeah, the FDA can issue observations, which are basically official warnings, and in severe cases, they can disqualify a testing facility, meaning their studies might not be accepted.
So GLP is really about protecting public health. It's not just a set of rules, it's about upholding the scientific integrity of the whole process.
Well said.
Okay, so we've covered a lot today. It's clear that good laboratory practices are absolutely essential for generating reliable data in non-clinical studies, and that data is ultimately what helps ensure that the medicines we all rely on are safe and effective.
Couldn't you put it better myself?
So for you, our listener, as you think about all of this, what do you think is the biggest challenge for labs when it comes to consistently maintaining these high standards? Or, looking ahead, how do you think GLP will need to evolve as technology changes and we see more digital tools and AI in the lab?
Those are some great questions to ponder.
Because GLP is only going to become more important as science and technology continue to advance. Thanks for joining us on this deep dive. We hope you've learned something new and interesting today.
And we hope it sparks some more questions and thoughts for you to explore.
Thanks for listening.